Buongiorno, grazie mille. Voglio parlare in inglese. Next time maybe in Italian. But. So the presentation, the title is Cherry Breeding in the World, Current Analysis and Future Perspectives. And just a very brief presentation. I work at INRA Bordeaux in a team called Adaptation to Cherry, of Cherry to Climate Change. So we already talked about this in the morning. And I have been I'm the breeder over there, let's say, since 2007. So before talking about methodologies in the field of breeding, I would like to recall, even if we talked about it and there will be other talks about this, but which are now the most important cherry cultivars. And here I present the results of a survey that we did when we prepared the book that Professor Costa showed this morning, thanks to him to make the promotion of the book. And actually, what we asked to representatives of many countries was to give us the list of the most important cultivars. And what we saw is that there is already a high between country variabilities, with countries that have listed or gave 26 names, up to Turkey, for example, where only cultivar Ziraat, as you can see, is extremely important, representing 70 to 80% of the production. Also, there were, well, at the end there were 132 cultivars that were selected with 1 to 14 occurrences, so being important either in 1 or up to 14 countries. There are other cultivars to be mentioned, like Bing, for example, which is very important yet still in the US, in Chile. And the case of Iran was also interesting, since among the 6 cultivars that were cited, 4 were local, including a cultivar named Sayed Mashhad, that we hypothesize maybe could be a synonym of Noir de Meshed. And in this list, data was missing for important countries in terms of production, such as Russia, Syria, and Uzbekistan. Here what I present is a selection of 10 cultivars that were considered to be, in this survey, important in at least 5 countries. And in orange, you can find self-fertile cultivars, very important, all originating from Stella. In yellow, a blush cultivar, which is Rainier. And in green, I put the countries that produced at that time at least 50,000 tons of sweet cherries. Also, it's interesting to see the case of Belge. Well, I put Belge because it's how we call it in France, but Belge actually is the same as Ferrovia in Italy, but also Gemmersdorfer in Germany, or Schneiders, and even Noir de Meshed. According to molecular markers, these cultivars, or at least the ones that we use in Europe, would be identical. So this is just to give a brief introduction about what are the most important cultivars still used today. And in this, when preparing this book, we also conducted a survey on the most active breeding programs, in particular the public breeding programs, because getting the information from private is not always so easy. But before giving some details, I wanted to point out that today cherry breeding is the second most active in Prunus after peach, with 230 new cultivars between 1990 and 2004, at least at this period. Maybe now apricot is more active, I don't know, but somehow it's very active research area. And in this book we describe 22 programs from 16 different countries. So here I just highlighted the one in France, which started in 1978, and the number of cultivars that have been released by each program. So in this book also we described the 119 cultivars which were considered to be of global importance or local importance and promising. And out of them, 80 more or less were created by public programs. And I wanted to highlight the importance of Canada and the program of Summerland, with a very long list, as you can see, of cultivars that are considered as very important in many countries, not only in Canada. And of course, this shows the importance that self-fertility gave them as a strategic advantage. So one of the conclusions that we can say about the situation of cherry breeding, like for many other species in fact, is that the genetic base that has been used is quite narrow. And this has been demonstrated in several scientific papers. And some of the reasons might be that there are very few sources of allelic diversity for traits of interest, like for example self-fertility, which is derived from Stella and then Lapins. Well, I put Lapins, but I could have put Stella. The tolerance to rain-induced cracking, with very few cultivars considered to be tolerant, such as Regina. Also, the difficulties in cherry breeding to create large progenies. We will see some figures about this. Because even though a cherry tree produces a lot of flowers, to manage to have a good fruit set when doing crosses is not easy at all. Some alleles for important traits, like fruit weight and firmness, are dominant. That means that if you cross two cultivars with quite nice fruit weight, many of the hybrids will have small fruit weight because there are some alleles over there that will combine in a way that they will dominate. And also breeders, during many years, they have put the weight of their selection in just a few traits. And of course, if you only work or you give too much weight to traits like fruit size, firmness, and so on, you will end up using a small number of varieties. So before giving some figures about hybrid production, I wanted to give you the detail of the structure of a sweet cherry breeding program. Like, for example, the one we conduct in France. Of course, that there can be variations. I will talk about it. But we start by the hybrid production. We have a year in which hybrids are often planted in a sort of nursery. And after they will be planted in the field and evaluated for 2 or 3 years once they have passed the juvenility period. So you can see we are already in years 2-9. Then we will select the best hybrids, which will be multiplied and evaluated in 1 or 2 levels before the pre-commercial trials and the cultivar release. During the phase of the nursery, it's where we can apply molecular markers and do selection assisted by markers. I will talk about this a little bit. This is what means mass there. So the selection can be based on genotype and on phenotype. And at the end, of course, a cultivar has to be registered, and this goes through these famous DAS tests, distinction, uniformity, stability of each new cultivar. These arrows here, what they show is that a breeder doesn't necessarily need to wait until his hybrids become a cultivar to use them as new genitors and to start creating new generations of hybrids. They can enrich the pool of genitors in any of these stages if the breeder considers that they have good quality. So this is a sort of recurrent selection cycle like it has been described in major crops. And of course, if you don't want to wait 30 years, you can and you have something very promising, you can, for example, skip this hybrid evaluation 3 level and go directly to pre-commercial trials. This will depend, of course, on the economic resources of each breeding program and the objectives. So another way of presenting this scheme is with numbers of hybrids that are created and are selected. And there is a bottleneck, like in any breeding program, and it's based on the law of big numbers. It's interesting to manage to produce large progenies, but we'll see that this is not so easy. What are the hybridization possibilities? Breeders can do controlled crosses or open pollinations. In controlled crosses, they can do hand pollination. They will put the pollen of parent B on parent A, or the other way around. But if they use, and this can be done manually, as you can see over there, if you use insect pollination in cages, like for example in this picture, and you put beside the tree that is planted a potted tree, for example, which flowers at the same time and the trees are compatible, you can collect fruit in both parents. So you can, in one cross, get the combination AB or BA. This is just the main difference. And in quantitative genetic theory, we say that this gives information on the specific combining ability, on the value, precise value to express it more easily of that cross. But if you do open pollination, which is easier, you don't need to do the crosses, you just collect fruit from one parent A, which will be pollinated by the sweet cherries around, you will just get an information on the general combining ability of this parent. But open pollinations have been used successfully by many breeders, and many of the varieties come from open pollination. So it's something that cannot be neglected. There is one last type of pollination scheme that has not been too much used, or at least reported in sweet cherry, is what we call polycross, where a series of parents are put all together, for example, in a greenhouse with insects, and everyone pollinates everyone, as long as you can see they are more or less from the same flowering period. So early flowering parents will pollinate each other, mid-flowering, late flowering, and so on, as long as they have different alleles of the system of incompatibility. And actually, if you take the fruits, for example, from parent A, you can have like a sort of general estimation of the general combining ability, which is restricted only to the three parents that can pollinate parent A. But if you use molecular markers, you will be able to know of the hybrids collected in parent A, which is a parent. Is it E, H, K, and then you can calculate or estimate the specific combining ability of all of these crosses. So this is just a little bit of theory, but let's go to real life. And this is, for example, the results of hand pollinations conducted at INRA from 2008 to 2018. And we have three different graphs. In the first one, we estimate the total fruit set. So out of the number of flowers that were hand pollinated, how many fruits we collected. So in 2012, 0. There was very bad conditions. But in general, we get something, but it can be quite low, and the maximum is 8%. Then when you get the fruit, you have not win the game yet, because you have to make them germinate. And here as well, you can see that some years the germination rate was relatively bad, and some others much more successful. It depends, of course, on the varieties that you used, on conditions. And at the end, when you combine the fruit set and germination rate, then you have the success rate, final success rate. That is, out of the flowers you pollinate, how many hybrids you produce, how many trees at the end. And as you can see, it's again quite variable, but the best year, we managed to get 2.6%. So it's quite kind of cruel if we think about other fruit species. Probably other breeding programs have better results than what we do, because the conditions in Bordeaux during flowering are not always so good. And maybe also we have been using difficult varieties, for sure. And this, we're going to see it here. There is wide genetic variability in terms of fruit set. These are the genitors, some of the genitors that we have been using. And you can see, well, for some of them, the crosses didn't work. Probably we didn't use them much. And for some of them, we had up to 20%. Again, for percentage of germination, the variability is even larger. And for example, we've used a little bit burlat, which is one of the earliest ripening cultivars. And burlat hybrids collected in burlat have very low percentage of germination, as you can see here, but also earlies and some of the early ripening cultivars. Whereas some of the late ones, we've have percentages up to 85%. And at the end, again, if we combine, we can class our genitors by overall success. And for example, just to give you an example, there is a variety from INRA called Fermina, this one, which is quite interesting for cracking tolerance as well, that we've been using quite a lot, but using it as a mother genitor is really bad. At the end, we don't know, it must have some fertility issues in our conditions, so now we stopped after all these years, and we collect pollen from Fermina and use it in the other way around. But you need experience and years to come to these conclusions. So now we'll move to another part of the work in breeding, which is, I think is also of interest for growers, which is the genotypic selection. That is the use of molecular markers to select individuals. So the first step is to study what we call the genetic determinism of traits, or, maybe easier way, the regions of the genome which control the variability of our traits. This is called the QTL mapping, but we don't mind so much about the terms. And then we have to go from this scientific discovery to the development of markers that can be used by all the community of breeders. So I quote a sentence from Cameron Peace, a colleague from Washington State University, who said a few years ago, or reported a few years ago, this sentence, that, that we assume that discovering a QTL is all it takes to then be able to conduct marker-assisted breeding, but then we realized that discovering a QTL is just the first step. It's much more complicated. And well, I wanted to put the pictures of these two colleagues, from Amy and Johnny from Michigan State as well, because thanks to some American projects that they've been coordinating, we've been able to advance quite a lot in this field. So just to explain very briefly in what this consists, what we need to have is a family with, for example, 120 hybrids. You will see they are all from the same mother and the same father. We will study the DNA of each of these individuals with molecular markers. This is what in that table I wrote M1 to M13, but we can have many more markers. And no matter what, the information of a marker is coded in a certain way. Here it's coded as a presence or absence, 0, 1. And then for all of these hybrids, we are going to measure phenotypically the trait. For example, C1 is fruit weight. So the first hybrid has a weight of 11, the second 6.1. And the trait C6 is firmness in a Durofel index from 0 to 11 to 100. Then with special statistical software, we are going to combine all of these, and it's going to tell us that, for example, in this chromosome, which is represented by this bar, these are molecular markers, there will be a region which is controlling somehow the variation of the trait. And this is the regions that are going to be interesting for us. So I made a summary in cherry of the main regions that have been so far identified for these traits that you can see here. So bloom date, maturity date, fruit weight, fruit firmness, cracking tolerance as well, and fruit acidity. So you can see there are many more, but these are the most important. And what's interesting is that in certain chromosomes, I forgot to say that sweet cherry has a basic number of chromosomes of 8, like all the prunus. In chromosomes 2 and 4, we have a high density of regions explaining the variation of these traits. So interesting genes at the end. So I will just give an example of this type of marker-assisted selection that is being implemented now, and it is the one for fruit size. So the team of Amy and Johnny in Michigan State University was the first to discover an important QTL explaining up to 30% of the variation of the trait fruit weight. And then through a collaboration, we discovered a potential gene that regulates cell number in the fruit that could be involved. But this is just theory. What is interesting for us is the applied work. And what was described later on by Cameron Peace was the use of the genetic information of these two molecular markers that surround this area. And actually, these numbers, it doesn't matter what it is. Actually, it's the size of a band in a gel, in an electrophoresis gel. But what we discovered is that some combinations of alleles at these two markers were associated either with larger fruit or with small fruit. And this is what the breeders we can do now. When we cross two varieties, we take the DNA, we do the PCRs and so on, and we can discard those hybrids that have unfavorable alleles that explain or that will tend to give smaller fruit. So what we did, for example, in INRA was to characterize some of these varieties, and then the combination of alleles with the two markers is what we call haplotypes. And well, the summary is that we can see which ones have favorable or unfavorable. And for example, if we cross Ferdiva and Fertar, which have both green haplotypes, so for both of them the alleles are favorable, we cannot do marker-assisted selection at this region, because these two cultivars have already the good alleles. But this doesn't mean that if we cross them, all of the hybrids would have big fruit, because other regions are explaining. So this is what we need to develop, markers for the other fruit weight QTL. So you see, it's quite complicated, but at least for this one, we already have tools for it. So the perspectives for this breeding work, first of all, I would say it's important to enlarge the genetic basis. I think we should more and more integrate land races, that is old varieties, and even wild materials to do what we call pre-breeding activities. So that is to create the genitors that have traits of interest, but that are still not good enough to become a cultivar. Of course, we have the problem of unfavorable alleles in these materials. We will have a lot of small fruit and other problems. Improvement of QTL detection, I will not enter into details. The work of translation from scientists to breeders. Also phenotypic selection. Professor Costa talked about NIRS this morning, but we could also imagine to use imaging techniques, improve the resistance evaluation, and so on. Something very important as well is the evaluation of genotype-environment interactions, and in general the adaptability of our hybrids. And in this context, to create networks between breeders and evaluators is a good idea, I think. And I wanted to say a few words about the impact that new pests and diseases can have on breeders' activities, for example, Drosophila suzuki. This is in Bordeaux. We have tremendous problems with Drosophila suzuki, but because we have 12 hectares of hybrids, each one with a different maturity, very big trees, and we don't have the same resources as maybe growers can mobilize. We cannot do what Professor Mori said this morning, to collect everything. And so we end up enclosing one branch with Suzuki-proof net to be able at least to evaluate correctly the quality of that fruit. So it's really a problem for us as well, and I hope better solutions will be found, because also the phytosanitary protection doesn't work when you have this type of orchard. And so, given all these challenges, there are three practical goals that we could try to go forward. First, of course, is to continue the intensification of collaboration between breeders and scientists. Also to strengthen the links between research institutions and the private sector. I think this is more and more the case, but it should be continued. And as was said this morning, but one of the political representatives, I think, also to improve the organization and the structuration of the sweet cherry sector, not only research, but at a higher level, at the European level. And with this last slide, I would like to thank you for your attention. This is two different pictures of our team in two different moments. I would like to thank also CEP Innovation, which is a French company of nurserymen who is supporting financially our breeding program. And of course, the fruit experimental unit at INRA Bordeaux, who takes care of our trees, and a series of projects. Thanks a lot.
Thank you, Jose. Thank you very much.